Autism and Public Health lecture, which I think will be our 8th or 9th, is coming up next month. Julia Bascom is going to talk about Nothing About Us Without Us, Autistic People Shaping Church and Fathers Raising Children on the Spectrum, coming up on Thursday, April 2nd in the evening, also here in Baracus. And for those of you who are from Drexel but not from the Autism Institute, especially I want to call your attention because I think we're hyping it pretty much in the Autism Institute quite a bit, but we have revived our Drexel Autism Consortium, and we'll be having the second meeting on April 6th over in the Skyview, and we're bringing lunch, so hopefully that will help people come. We're going to do one more rapid research rounds type of meeting where people get to take a few minutes to just talk about their work and their interest in autism. And then after that, we'll be breaking out into small groups for the next meeting to start to formulate ideas for new collaborations. But it's my great pleasure to welcome everybody to hear Dr. Kevin Pelphrey today. He's the Harrison Wood Jefferson Scholars Foundation Professor of Neurology and also holds secondary appointments in the Curry School of Education and the Department of Psychology at the University of Virginia. His training has been mostly in that Middle Eastern region. He first got his bachelor's degree at North Carolina State University, his Ph.D. at UNC Chapel Hill, and a postdoctoral fellowship at Duke University. He has a really illustrious career, and I cannot possibly summarize all of it, but just to mention a few highlights. In 2013, he founded the Center for Translational Developmental Neuroscience at the Yale School of Medicine. In 2016, he founded the Autism and Neurodevelopmental Disorders Institute at the George Washington University and Children's National Health System in Washington, D.C. He has a whole page of awards and honors. Just to mention a few, he's been identified by the American Psychological Association, the NIH Interagency Autism Coordinating Committee as an outstanding scholar and somebody who can really represent our science to the community. He has more grants than I can count. Actually, I tried to count, and I think I exceeded 25. So several of them are current, and it's really interesting to see as I kind of look through some of your body of work. What I first knew you for is some of your imaging work on social perception, social cognition. He studied tasks including face processing, audiovisual integration of speech perception, eye gaze, biological motion. But most recently, what really got me excited to bring him here where our focus is more on public health science is to hear about some of the work that he's doing, trying to better understand what females with autism are like, both from a neurodevelopmental and neurogenetic perspective. And I'm really excited to hear about your work today. Let's see if that works. Building Precision Care for Autism, or Autisms. Thank you and welcome.
I'm delighted to be here. Thank you so much. I think you all are building an amazing autism program, and I was really excited to see the panel discussion focused on dads of kids with autism. I'm a parent as well as a scientist, parent of 5 kids, 2 of whom are on the autism spectrum. So our house is a really interesting place most days of the week, and I'm delighted to be here. So, the unifying idea for the talk today is this focus on how we define autism and the notion that given that autism remains a behaviorally defined disorder, so when we think about autism, we think about a cluster and a set of symptoms and characteristics, but we don't yet really have an understanding of underlying biological mechanisms, and as a result, we don't really have a blood test for autism or any sort of sophisticated, mechanistically informed biological marker. And I would argue that this is one of the hindrances to being able to do what other fields of medicine are starting to do in terms of providing much more directed precision interventions and care. And so I want to talk about that, and when we think about both with my kids and kids that I know professionally, the way they go about kind of receiving interventions and the types of interventions received are really more about the happenstance and chance meetings of different clinicians, what's available in your particular state, what university do you live near in terms of which behavioral intervention is the one your child is most likely to receive, and it's not informed necessarily by a real understanding of individualized characteristics. And so I'll talk about that a lot today, and particularly in the case of girls with autism. I like this slide. This has nothing to do with autism, but it was in an article on precision medicine. It's titled In Precision Medicine, and it stunned me. As a psychologist, I think I always perceive other fields of medicine and healthcare as being much more sophisticated than they really are. But this is different drugs, and many of these are, kind of the point of this is that these are blockbuster type drugs, so Abilify, Nexium, Crestor, and what they're showing is the number of people that you have to try the drug in before you get somebody who responds positively, for whom it works in terms of the criteria you're trying, or the symptoms that you're trying to reduce. And that stunned me. For example, Crestor for high cholesterol, you're giving it to, looks like a little more than 20 people there before you're identifying one that's a true responder. Very much surprised me, especially when you think about a lot of behavioral interventions, cognitive behavioral therapy as well, that we've developed in the field of psychology, where we tend to think about half of people are responding quite nicely. So I was actually sort of proud of my field there after seeing what I always perceived to be the much more mature fields. So certainly we're not alone in this idea of precision versus imprecision medicine. But the thing about, of course, with the exception of schizophrenia, depression there for Cymbalta, you've got conditions that in, I guess most people's opinions are very well defined, and for some, there's a clear blood test, high cholesterol, for example, for Crestor. And so, that area, that aspect of envy is still there. So, as Diana mentioned, a lot of my work over the years is focused on a really simple aspect of what human beings do as they navigate the world, and that is determining what's another person, what's a social agent in the environment versus what's not. And it turns out that even very, very young babies do this pretty much the day they're born. They're able to distinguish between things that I'm showing you here, biological or social motion, versus scrambled or non-social type motions. And these are just made by monitoring little infrared light reflectors on the body. And so this is a colleague of mine who was playing patty cake at the time with the camera, and this is the scrambled version. So we've made a lot out of these types of stimuli because they do a really good job of differentiating between brain systems that are involved in processing other people and brain systems that are more generally involved in processing different types of motion and different types of physical aspects, the physics of the world. And I think it actually bears pausing there for a minute. It's a fairly, I forget this at times, that it's a fairly new area of neuroscience that we think about in terms of talking about social neuroscience or the social brain, terms that you've probably heard for many years now. But when I started as a postdoc back in 2003, 2004, that was a really new area, and it was widely debated whether or not humans sort of had this dedicated machinery for processing social stimuli. And now it's much less debated. The kind of the Bible of neuroscience, the neurosciences textbook now has a chapter on the social brain. So it's in there in the same kind of level as language processing, higher level reasoning, systems for reading. And that's exciting to see, and it's particularly exciting from the point of view of trying to understand autism, in that one of the defining features of autism is this qualitative difference in social cognition and social perception, sort of social being. And so we've always thought that that must be related to differences in the social brain, and that seemed like a very reasonable place to start, and that's kind of where I started my journey in terms of studying autism. So I want you to kind of keep in mind these stimuli, and I'll show you a few other sets of data where we've tried to do very particular things with brain imaging. And one thing that we've tried to do, so the field of imaging, using mostly functional magnetic resonance imaging, as applied to autism, started out looking to define group-level differences between people with autism and people without autism. And usually the people without autism were kind of an unusual group because they were neurotypical, so everything was excluded that you could have in terms of sort of neuropsychiatric disorder, which is sort of, that's a very particular type of person. Most of us have a little touch of something. And so it was, in a way, an easy problem to define what's different about a group of people with a very clear behavioral syndrome versus a group of people that have no such conditions. And that led to studies that very nicely kind of started to illustrate the neural circuits that, on the one hand, could be driving autism symptoms, and on the other hand, and fairly equally likely, reflecting what happens to you if you have autism and you're living in the world. Because most of those studies were done with young adults, just as a sample of convenience. But even when those studies began to be done with kids, what you're seeing is these group level differences. And that transitioned really over the past 10 years to trying to do something much more individual specific. And so it's a different problem to say that if you have a group of people and among those people an individual has autism, and find the individual from their pattern of brain activity that has autism, and what does that look like? That's a different problem, and it's a more challenging problem. And then an even more challenging problem that's related to that is saying something about an individual's level of social or autistic symptoms on the basis of their brain scan. And that's really what we would like to be able to do if we want to start having a biomarker as we commonly understand it. So back now 4 years ago, we published a study where we were looking for the first time for our group, and then surprisingly, it was really the first time in the field, which I didn't realize this until we started publishing it and reading the other papers very, very carefully, where we were taking a discovery sample and then we were using machine learning techniques to look at our fMRI data and say, how does the machine learn to differentiate, how does our algorithm to differentiate between people with and without autism? And what we found was that we could then apply that technique to a new group, a replication group, where we had people with or without autism, and begin to actually predict the cases, okay? Two things are really important to remember about this, or to note. One is that our problem, again, was a fairly easy problem. Like, the clinical problem isn't, does this person have autism as a child or have nothing? It's, this person has some behaviors that are concerning and limiting their ability to adapt in the world. Can we understand a diagnosis here so we can make some predictions and understand, so it's a differential, of course, right? So it's ADHD or autism, is it this or that? So we did a very simple problem, and this is a criticism both of our study, but also the entire kind of field going back. Whenever you see these attempts to use machine learning, artificial intelligence techniques, when, I'll show you some receiver operating characteristic curves, understand that prior to this study, none of them had applied their algorithms to a new sample. What they were showing is how well in the sample that it learned on could they then differentiate the sample. And that's cool. It's a good way of showing an effect size, but it's not the same thing as making a diagnosis, nor is what we're doing, because clinically this is the easiest thing in the world. Any of the clinicians in our group could have done the same thing the computer did without the $1 million for the magnet and the expensive scans. So it's a very simple clinical problem. But nonetheless, we were able to, in most of our subjects, do a really good job of saying which have autism and which don't. And what I want to kind of walk you through this. So panel C here is showing how well we could do in terms of the boys in the sample using these little regions of the temporal lobe called the fusiform gyri and the superior temporal sulcus, which are part of a network that processes the social motion that I was showing you earlier. So we could do a good job of differentiating boys and girls, but if you're used to looking at these types of curves, you probably already noticed that in this one, we're doing really poorly, right? And this is girls. This was the first study that we had where, by chance, we had collected enough girls with autism to be able to break out and kind of look at them separately. And so in terms of transparency and honesty, even though we wrote the paper like we had that idea all along, what really happened was that we had all the sample together, and boys and girls, and all of a sudden what we had found for many years, that these kind of social brain regions were good at being related to autism, wasn't true anymore. And we're like, well, this is the largest sample we've ever had. Maybe that's the issue, that when we're studying it in a large group, the effect goes away. There's false positives and rejecting hypotheses falsely, that happens. But we didn't want to settle for that. And so we took the girls out, and all of a sudden we had this beautiful effect that we had seen in many studies come back. And then I said, okay, wait, put the girls back in, and it goes away. I'm like, well, plot the girls. And, well, that's actually below chance. So the social brain network that's really good at identifying boys is not only bad at identifying girls, but sort of counterpredictive to predicting autism in girls. And another way of saying that is, if we were to look separately at a large number of girls, chances are they would have a robust social cognition, social brain system, even though they have autism. And this was stunning to me as somebody who was sort of a true believer in this kind of social brain idea in autism, because I would never have predicted that there were such profound sex differences, especially when everybody that's in this study has gone through the ADOS and the ADI, has seen a very experienced clinician first, so clinical judgment informed by ADOS and ADI, and has had, because of their age, usually a couple of other clinicians that have seen them over the years. So these are rigorously characterized individuals with autism. And if anything, we should have been making them much more like each other. So, as we started talking with colleagues who were very familiar with girls with autism, and as a parent of a girl with autism, there was sort of this idea, but wasn't certainly backed by imaging data, that the girls were different. And one way that they were different was that, these sort of core social symptoms were different. It wasn't until you really pushed into more sophisticated aspects of social cognition that you could see differences. And oftentimes that was what was sort of naturally happening in middle school. But for boys, that kind of classic picture of a lack of social motivation or a lack of interest, and but that wasn't true of girls. And that's exactly what we seem to be seeing here. So we published that finding, and at the same time, we were going after an Autism Center of Excellence network that was focused on girls. So it wasn't just that set of findings, but this broader idea happening and coming up more and more in the literature, that girls were being missed. And that was certainly my experience. My daughter wasn't diagnosed until about the age of 4.5. And in contrast, for example, I have a son who's diagnosed with autism. He was diagnosed about 16 months, so very, very early. And a lot of the delay for my daughter was kind of repeated occurrences where clinicians thought, well, maybe, but she's social, she's interested in interacting, but it's different, and so sort of this delay in diagnosis. And that was a very common occurrence, even for people like myself, a family that's in the field, so we had access to the very best people, but still this delay. So I couldn't imagine what it would be like for a family with less resources and less of a connection to those particular resources involved in the autism community. So as we looked at this, it's obvious that we have girls are less likely to meet the diagnostic criteria, more likely to get a series of inaccurate diagnoses, which can be really damaging in terms of the different attempts at treatment that can be counterproductive. Oftentimes, for a girl to get the diagnosis, it requires an additional mental health problem that gets them in trouble and is annoying to people around them, is disruptive in school. And so, there again, missing that opportunity for early intervention. And then something that's particularly concerning is the greater risk for victimization that comes with both a misdiagnosis, but also even an accurate diagnosis. Girls with autism are at greater risk for victimization in school. So this is just a cartoon of our network. And so now we've got West Coast sites, including San Francisco and UCLA, Seattle, Washington. We're in Charlottesville. We have a site in Washington, D.C., and then at Yale and at Harvard. And what we're doing is collecting imaging data. We're collecting EEG, genetic material to do whole genome sequencing, blood to do gene expression, very careful assessments of different aspects of cognition in the phenotype. And originally, we started this out studying girls who were between about 7 to 17, actually girls and boys, I'm sorry, about 7 to 17 years old. And so we recently renewed this, or say recently, about 3 years ago now. So now we're in the middle of collecting those same research participants, but at a later time point. So there's a really cool opportunity to see for all of these girls and boys, as well as unaffected siblings who are brothers and sisters, we're able to see that transition through puberty into young adulthood and then into kind of going out into the world out of educational experiences. And that's a little bit of what I'm going to tell you about today. One of the things that, and it's really cool to see that you have Julia coming, because Julia is actually a collaborator in our ACE network. Her Autism Self-Advocacy Network group are scientific colleagues with us on this. And what we did, we realized that while we were studying fairly young kids in our first wave, in the second wave, our kids were old enough now to really, tell us what it was like to have autism, to talk to us, as young adults about their experiences. And this has been really cool. She will probably share with you some of the data from this, so I won't spoil that, but I think it's very fascinating, and it's a new thing for me because I'm used to very, simple biological studies where we're looking at a brain system and maybe we relate it to a treatment outcome, but that's as sophisticated as it gets. With this, we're doing these really cool cognitive interviews around the process of getting an ADOS, and like what's that like, and asking questions about the questions, and with young adults. And one of the things that's come up, a new phenotype, just out of the discussions that we're having, that we're calling autistic inertia, this kind of element of executive dysfunction, but related more to a motor hypotonia and kind of being out of touch with one's inner workings. And so, we wouldn't have known to as a neuroscientist, that fascinates me. That immediately suggests several brain systems that I've never really thought about in the context of autism before. And so, but we wouldn't have thought of that without working with our colleagues who have autism. And so not only are we working with Julia and her colleagues in that group, but also then identifying research participants who want to do this work with us, where we're doing the cognitive interviewing and we're actually developing measures of autism symptoms that are focused on young adults with autism based on our conversations with those individuals with autism. The other thing that's very new for us, so in the original Wave 1 data, we were going based on genetic sex, so just chromosome X, XY, and here we had the opportunity, because of my colleague, Dr. John Strang. He has a clinic that focuses specifically on gender diversity, and it's sort of a chicken and egg question in the sense that the clinic, he's also an autism expert, and so he was getting quite a few people with autism coming to him anyway, but then when he opened the gender diversity clinic, he had many cases that were coming to him because of questions about gender diversity, but then it was clear that his autism expertise was very relevant based on the presentation of the individuals coming to him. And so he started thinking about, well, what's autism, what's this concept of gender dysphoria, how do these two interrelate? And he has a K award focused on this now that has added several measures of gender diversity, gender dysphoria, better measures of gender identity to our sample, so that we not only have the genetic characterization of biological sex, but an understanding of gender identity going into that, and how that relates to our the development of our young people who are facing both living in a world with autism, but also having gender differences, and whether or not their social network is supportive, and who they have to talk to, and how they're handling any transitions that are happening. And so it's a very, I think, exciting opportunity that gets us delving deeply into some just really fundamental aspects of defining who our participants, defining who they are. And I find that fascinating from a scientific perspective. But also I think that there's very little work in this area, in an area where there's a lot of need and a lot of misunderstanding. And so we're trying to contribute to that and kind of give you the high-level vision of that. But I highly recommend you read John Strang's work. He recently published some guidelines around gender and autism and the intersection. So, okay, kind of coming back to a potential biological mechanism. So I've noted phenotypic differences in autism, and I've noted that certainly girls with autism receive diagnoses later and fewer girls are diagnosed. I've kind of treated that as though there wasn't an underlying biological reality necessarily, but it's important to say that probably the most replicated biological finding in autism is the difference in sex ratios. So, depending on how you're defining autism, anywhere from 8 to 1 to 5 to 1 in terms of more boys than girls, and that's really interesting. What's going on, or it can't, I don't think it is all identification and ascertainment. I think there's an underlying protective aspect to female development, female brain development. And kind of what that is, I think, could be very important to understanding broadly. How do you mediate risk for autism? And what is it about developing as a female that allows you to avoid that autism phenotype? And so in the genetics world, this is often called a Carter effect. nothing to do with politics, but it's that you have to have a stronger genetic hit to get to that phenotype if you're a girl versus a boy for a number of reasons. Often the reasons are very vaguely referenced, like differences in hormones, differences in underlying patterns of development. So that's shown up very clearly in the genetics of autism. And even though we are very tuned into differences in ascertainment and diagnosis, we still believe that there is this underlying difference in genetic liability. And so we've been exploring that with our genetics and imaging data. So what I'm going to show you now is an attempt to enrich what we can do with imaging. And I guess it's also an attempt to enrich what we can do with genetics by really trying to combine them in what I hope you'll think is a clever way. So the simplest thing that we did with the imaging is to replicate that prior finding that I mentioned about the differences in neural systems that are related to the social symptoms in autism. And so what I'm showing you here, this is simply with a large sample of boys and girls with autism and typically developing boys and girls. What I'm showing you is the difference between typically developing girls and girls with autism, and that is the blue activation maps here, okay? So these are brain systems that reflect the diagnosis of autism compared to typically developing girls. The red is the difference in brain systems that characterizes autism in boys versus typically developing boys. And these hold true also when you compare to opposite sex and across diagnostic conditions. So I'm showing it this way for simplicity. And this is during that processing of the social motion that I was showing you earlier. Do I have a question? Oh, sure thing. So this is how deep you are in the brain if you were slicing from the side, okay? Yep. Yep. So what we're showing is how far in the brain you're going. And this is from the side, so what you're seeing is the temporal cortex right here in the right hemisphere. And what this says is that there essentially is no overlap between the neural systems that are affected during social processing in boys and girls with autism. And so even though, again, they've all been given this rigorous diagnostic criteria, which is a bit of a, not a metaphysical question, but it's a philosophical question for us. So we've given them the diagnostics, and we as a group, and I think a field, strongly suspect that maybe the diagnostics are a different experience for the boys versus the girls, but assuming that they've all gotten the same diagnosis and therefore all kind of put through this filter. So again, their brains should be maximally similar in terms of neural system function, but nonetheless, the boys versus the girls have non-overlapping affected neural systems. So I mentioned before that with this type of design, you can't say whether it's cause or effect. So it could be that the autism is affecting the boys and girls differently and therefore affecting different brain systems. Or it could be that these are somehow causally related, and it's like with everything, it's both, probably. And so it could be that there are different underlying causes in terms of the neural system manifestation of autism. Question? This is during... Yep, exactly. Mm-hmm. Yep. Yes. In terms of eye movements, which is what we have as a behavioral. Yep. So they look the same amount. What they're thinking about while they're watching those stimuli, we don't know. Yeah. But you're actually setting me up perfectly for the next study I'll show you. So, different overlapping neural systems. So then we took that data, so we have maps of where in the brain you're different, depending on what your group membership is, both by diagnosis and by sex. And we overlapped that with gene expression maps that are available from the Nod Sestins lab that allow us to say what genes are expressed in different parts of the brain at different points, mostly in fetal and very early postnatal development. And we created gene sets based on that. So this is the overlap between gene expression and brain differences. And then we looked at that in terms of the overlap with our whole exome sequencing that we had done. And what we found, on the one hand, we replicated an older finding, not very old, but it hadn't been replicated before, where we showed that you needed bigger and more copy number differences in terms of genetic hits. If you're a girl, the girls had more and bigger of those in order to get to the same behavioral phenotype. So remember, I kept emphasizing same behavioral tests. And I should have told you that in terms of autism severity, very well matched. And in terms of IQ and all the different variables you would want to match on, boys and girls are very well matched. So what this is showing is they require a bigger hit. And furthermore, when you look within the neural circuits that we were defining with our activity maps, and then the gene expression data, that when you narrowed it to a search within those regions, you saw very clear differences in terms of the types of genes that were hit within those networks that were making up this broader data. And it looks like, at first analysis of that the networks for boys are very much related to very well-accepted ideas around synapse development, synapse elimination, kind of, pure neural stuff that people have been talking about in terms of autism for years. But for the girls, more clustering within inflammation, especially handling early information in glial processes in autism. In prenatal development. So it's suggesting that we've got different brain systems, different numbers of hits you have to get to the same behavioral phenotype, but different brain circuitry, and then different pathways of genes, depending on whether you're in the boy or girl group. And so, it's looking more and more like these are really different conditions in terms of underlying mechanisms, even though, again, I keep emphasizing, gold standard diagnostic instruments say that you're not seeing any differences between the boys and the girls when you're looking at getting to the diagnosis, right? Which is a very weird group. That's kind of a very research-specific group. So the reality of this is probably even both messier and bigger effects when you're looking at this in the real population of individuals out there. Yep. Mm-hmm. 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 I think we want to dissociate between those two hypotheses. On the one hand, this could be there's something that we're not studying that would tie these differences together. And then on the other hand, these are two different disorders, even though we've been calling it the same thing. And that then begs the question of, well, what is, if you had the genetics in the brain system that would make you a boy with autism, make you look like the boys with autism, what do you develop if you're a girl? And like a pet theory of mine is actually that's a pathway towards anxiety, depression, and eating disorders is my current kind of working hypothesis. No real data for that. It's just working hypothesis. Actually, a little bit of data. I'll show you in a second. Okay, so your question about you're showing them biological motion stimuli, and then you're getting these brain differences. The problem with that is it's sort of prejudging what you're using as far as the stimuli to probe. So you're already putting a theory in there. So what if, it's just that social information isn't the best thing to use? So here, what I'm showing you is resting state data, which is literally what it sounds like, putting the subject in the magnet. You're taking data on kind of co-fluctuations across brain regions. And then we're using a latent approach, a machine learning approach, where we're pulling out latent networks that best differentiate boys and girls with autism. So you think of it as like the boy autism network, girl autism network, after we do the different comparisons across those with and without autism, boys and girls. And what we pull out when we do this is networks, got the two different networks. Then what this is, this Wordle, is that we're saying, all right, we've got the networks which give us regions of the brain that best define each group. Now what we want to do is go to the literature, and neuroimagers have made this handy. There's something called Neurosense, and it's high-tech phrenology. You say to Neurosense, I have this XYZ coordinate of brain activity. Tell me how the literature has talked about this brain region in terms of papers published. And so, if you have one that's kind of in dorsolateral prefrontal cortex, it's going to tell you working memory, executive function, high-level reasoning tasks. If you have one that's in the posterior superior temporal sulcus, it's going to say biological motion, social perception. And it's completely data-driven. And then what I'm showing you here is the magnitude of the correlation, the effect size, between the construct that kind of gets spit out, that does get spit out of Neurosense, versus the points we put in. And for girls, the constructs and the size of that effect size is correlated or represented by the size of the font here. So emotion regulation being the biggest one here, person perception being the biggest one here for girls versus boys, and the boldness of it. So the constructs, the three top constructs, anxiety, emotion regulation, comprehension, reflecting the brain networks that we saw in the resting state data. So here, this is what they're doing, just lying in the magnet. For boys, the brain systems that are coming out are differences. And for both of these situations, we're talking about reduced connectivity in these brain systems and then putting it onto these constructs. So for person perception, comprehension, so one overlap, which is comprehension, language, person perception, social perception, very much the core characteristics of an autism diagnosis. So the boys, if you had shown me this a few years ago, just the boy map, I would have said, yeah, wow, it looks like you got out a kind of a network related to autism. Emotion, anxiety, comprehension certainly reflect my experience of a daughter with autism, but those aren't words, maybe comprehension, and maybe when you get into language, which is number 7, that is in the DSM for autism. So the networks, the brain is telling us that the networks affected in autism in girls are very different from those affected by boys, and not networks that I think are really captured by the behavioral diagnosis, which is fascinating to me from a naive treatment perspective. So it's suggesting that you could affect, you could go at this in a very different direction, but still treat core aspects of autism in girls. And then, of course, some of the approaches we already use might be still very relevant for boys and supported by this brain data, okay? That's going beyond the data, but it's intriguing to me as a possibility. So I want to take you, next to the last thing I'll show you, into really thinking, could we start to use an approach like this kind of biomarker approach to predict which kids would benefit from particular types of treatment. So I want you to put aside what I've told you about boys and girls for a minute, and we're going to focus on boys here. And what we've been doing, we've been working on, this is my colleague Pam Ventola, and she and I have been doing, she's a clinical psychologist, in an intervention trial where we're testing, we're using pivotal response training or treatment as a form of behavioral intervention that's evidence-based as a probe that allows us to understand brain mechanisms in autism. So a problem with the imaging literature is that it's all been very correlational, and you can't inject or do brain lesions and see what happens in human models, but you can deliver an evidence-based treatment and then study the effects, and so that's essentially what we've been doing. But in this case, what we're doing is taking a brain scan. In this case, it's in response to social perception, a social perception task, before the treatment and then one after the treatment. And then doing this pivotal response training. And in this case, we're using the social responsiveness scale as our primary behavioral outcome. And the black lines are the individual treatment patterns. So if pre-time point, post-time point, treatment for 16 weeks, and moving down is a good thing in this particular measure. And what we're showing is that for some kids, these are the individual children, the black lines. Some kids, this helps. For some kids, there's no real change. And for some kids, it gets a tiny bit worse, probably not significantly, and the red line is the average. And so on average, pivotal response training on the East Coast works as it does on the West Coast. And so most of the studies have been done previously on the West Coast with PRT. And so that's great, behaviorally, this works. But what about this variability? Because if insurance isn't covering this, or in our case, we're doing it for free because it's a research study, it's really expensive. And more importantly, it's time consuming. I mean, essentially, one parent has to get the child to, close to 40 hours a week, or make the child available at home for close to 40 hours a week. And if there's only one parent, who's going to be working to pay for the treatment? And even in the states where insurance covers it, they don't cover it very well. And so you've got a real problem, potentially, where for about half the kids, this isn't going to be very beneficial. And, who are they? And so we did two approaches. One was to use and poll our best clinicians at the Child Study Center and say, look at these profiles and these charts and observe the kid for a few minutes and tell us if you think they're going to be a good candidate or not. And then another approach was to take all the behavioral data we had, all the different measures of intelligence and cognitive ability, language, and use that, and also use machine learning to try to combine all those measures optimally. And then the third approach is brain imaging. So we looked at the scans prior to treatment, and we said, what are the neural systems that predict treatment? And I love doing this. How many of you are clinicians? Awesome. It's going to be real fun. So I'm going to tell you something that's going to make you roll your eyes, if I haven't already. This is a brain system involved in figuring out if something's social or not, and it's very predictive of whether or not a child is going to benefit from an intervention. This is the correlation plot with how much a child benefits versus activity and connectivity in this system and these other three. Wait till I tell you what the other three do. So this one's a nice parietal system that's involved in distributing attention. So if you're a child that has a robust system neurally for distributing attention, you're a good candidate for pivotal response training, especially if you combine that with a system, which is this guy, involved in emotion regulation, doing things like sitting and staying on task and tolerating interacting with somebody else for a while. And then this last one you're going to love as clinicians. This is reflective of how much you find social stimuli rewarding and you're willing to work hard to get more of them. So it turns out that neuroscience can tell you that if a kid can sit still, pay attention, and find working with you rewarding, that they're going to benefit from a behavioral intervention. And this is the part where everybody's sort of like, great, so how much do those magnets cost, and how many kids can you get through, and do you really want me to give a kid a scan and then believe the data about whether or not I'm going to give them 16 weeks of behavioral intervention? So it might surprise you if I tell you that all of the clinicians were worse than chance at predicting which kids would benefit. Especially one of them who's super, super famous, who was worst of all. I won't name names, at the Child Studies Center. And he still doesn't believe me. There, I really narrowed it down. And so, his name rhymes with mommy. And which is actually a really common finding in cognitive psychology. The more of an expert you are in a particular domain, the worse you are at making a prediction about any individual occurrence. And then all of the behavioral data, which takes even longer than the scan, even if we can put it together with machine learning, we couldn't find heads or tails of the neuropsych data that would predict which kids would benefit. But this brain scan could. Now, I'm still not suggesting that everybody give a brain scan and then start to give treatment based on it. But what I'm suggesting is that this begins to be a very useful quantitative way of capturing aspects of a brain system that would be predictive of treatment response. But even more, like if that's not enough, what I think is cool about this is that it gives you a map of brain systems where literally you're saying if there's high connectivity and activity between these regions, then that person will benefit from this intervention. So what we're doing now is combining that information with what we know oxytocin will do, intranasal oxytocin. So we've done a study where we gave just a single whiff of oxytocin, placebo-controlled, and we compared individuals on placebo versus the oxytocin. And in kids with autism, we could bring about more activity and connectivity in a map that closely overlaps with the map I was showing you in terms of treatment prediction. So what we're doing now is predicting that the kids that we give oxytocin to prior to PRT, if their brain system is reflective of one of the kids who should not benefit from the PRT, the oxytocin will benefit them more than the kids who would have done well in PRT anyway. Oxytocin won't hurt their performance, but it won't help those kids. And we're hoping that that will illustrate that we can use, in this case, a drug intervention essentially, given prior to a behavioral intervention, to up our chances of improving the response to the behavioral intervention, but most importantly, predicting which kids. So then the last thing I'll show you is, all of this seems fine, but I don't think I said that, really everything I've shown you is about 7 years old or older. When we can stretch it down to about 5, actually the PRT data, it was mostly 5-year-olds. But any younger than that, which is really where behavioral interventions happen for most of our kids with autism, this isn't practical. Getting them in the scanner, getting good data, it's very challenging. So I like showing this because these are my 5 kids. So on one hand, I get to show, I need an updated picture because that one's a few years old now. This is the car I wish I had, but I have these. And so I don't actually have this, but I want a Vanagon because that's the coolest car I'm going to get until they're all gone. And they're bringing those back, by the way. So, how do we begin to do this in a practical setting? And one of the things, we've tried various things. Certainly, I'm making the point here that we can do fMRI with babies. It isn't as impractical as I'm making it out to be. But one approach that we've been taking, this optical imaging approach, looks like it's going to be very fruitful. And one of the earliest things that we're doing playing with this is looking for differences in optical imaging predictors. So this is a measure that's much better at handling kids who can move around and also more naturalistic interactions. And so this is just data from a study where a student of mine was predicting language development from early infant-directed speech. But the reason I'm showing you this is that we're starting to move into a position where we think we'll be able to apply this at a very early age. And the study that we're getting ready to launch now is trying to actually apply this to a more population level screening approach, where we're saying that, we already do hearing tests with every newborn. Can we get an optical imaging device in there and begin to play these speech sounds, which we know activate many of the same systems that I've been talking to you about in terms of being predictive of an autism diagnosis, or correlated with, and also predictive of treatment response. Are they actually predictive of ultimately developing autism? And I think something that's unique about my position at UVA, one of the reasons why I was keen to move there, is access to the maternity ward there, but also the maternity wards for the ANOVA group, which is in Northern Virginia. And so that's an incredibly diverse swath of country. Probably don't think about it being, Virginia, but Northern Virginia has large populations from just about every country on the planet, because that's where people can afford to live if they're working with or related to the embassies that are in DC. And so you have an amazing population that's diverse. And I think that one of the things which I'm speaking to the choir here, but most of the neuroscience that has been done in terms of developing early screening or early identification has focused exclusively on infant SIBS. And really, if we're going to develop anything that might approach being useful in the population, then we need to be studying many more shades of risk for autism, because that's a very specific form of risk, and it's not reflective of what's the other common risks. I'm always careful when I say this, but if you think about it, the NICU is the number one environmental risk for autism, right? And now you see why I'm careful of saying that. I'm not saying the NICU causes autism, but when we look at increases in the incidence of autism, one factor is that we're really good now versus 10 years ago at keeping babies breathing, but we're in our infancy in really figuring out how to foster early brain development. So one of the things that we're very interested in doing is using these measures both as early screens potentially, but also as ways to assess early social brain development, and then apply that in the NICU setting so we can begin to design better environments for promoting brain development early on. So I'll stop there. Hopefully I still have time for some questions. Thank the funders of this work, and most importantly, thank my colleagues. This was definitely a team science effort, even though there's just one of us talking today.
You matched your girls and your boys severity of autism and intellectual development. Do those differences hold up, gender differences hold up, as you look at a more severe population or intellectually disabled population?
We don't know yet. We don't know. One thing to add to that answer is that the other place where we've really been surprised by our brain imaging results, when for many years, the imaging literature, us included, we studied what people call high-functioning autism, kids who could get into the magnet. And we made the argument that reflects pure autism. It's a convenient argument. But when we stretched those boundaries and started to study kids, for example, kids with profound intellectual disabilities and autism, of course, it's much harder to get them into the magnet, but what we're finding is that the brain systems there, again, are very different in terms of what's kind of the prime suspects. And we thought, well, it'll be very similar, but worse. No. So, for example, these social brain systems that are so good at characterizing high IQ boys or average IQ boys are perfectly well functioning in that population. And that, again, stunned me, especially coming from kind of my theoretical perspective. That's not what I predicted at all.
Hi, I was wondering if you had looked at genetic factors within families, like multiple generations of different genders, and if those pathways are similar or different.
Not really. The genetic data I showed you was a combination of simplex families, because a lot of them were also in the Simon simplex collection, and then multiplex families, which is obviously the more common kind of presentation. But we haven't specifically looked at multi-generation families where autism, girls with autism runs in the family. It's a fascinating group. That's actually my family characteristic. It's kind of the opposite of what you often see. The people who are affected are more likely to be girls. And my sister was a math major and her Ph.D. in math, and she likes to remind me of that constantly. Whereas I was a very poor student in that analytic area.
I'm wondering if you could talk a little bit more about the kinds of new contexts that you're envisioning that you would create in the NICU or for people on the spectrum. Like what is this new ecosystem you're imagining?
Yeah, so it started out being so I majored in architecture as an undergrad and psychology, and I wasn't a good architect. So originally it was like, we can create environments that are more beneficial. But lately, I've been working with a gentleman named Bill Petrie, and Bill is a fascinating guy. He started his career as an infectious disease expert, and he's responsible for figuring out a way to prevent the most common form of diarrhea in Bangladesh. It's like, wow, that's real science. And now he's turned his attention to cognitive development. He's interested in the microbiome and cognitive development. And so now what I'm getting increasingly interested in is sort of these non-obvious environments like the microbiome. And so we've got a bunch of microbiome measures and some ideas about how that relates to inflammation. And my NICU colleagues, their expertise has been for a long time in predicting and preventing infection. So they're very open to this, but they haven't applied that before to cognitive development. It was really, if they did, it was a brain infection. It wasn't these sort of subtle cognitive developmental differences. So that's one area where I'm fascinated by the very non-obvious things. I use that term. I don't know if people know what I mean by that other than the obvious, but I had a mentor as a graduate student named Gilbert Gottlieb, and Gottlieb was famous for manipulating the early experience of ducklings while they were still in the egg, and he showed that in contrast to what Konrad Lorenz had demonstrated or thought he demonstrated, that for ducklings in particular, but a lot of birds, it's the experience of their own species typical vocalizations that drives the attachment and imprinting process. And if you took away by injecting glue into their vocal cords, that self-directed activity, you could play the same activity and they could hear it all day, the mother could keep calling, but it was really that self-directed activity that mattered. And he called that non-obvious causation and had an epigenetic theory back before epigenetics was cool or mechanistically understood. And so that's what I mean by non-obvious things. And in the autism field, it's easy to kind of compare that to what people have thought of as obvious environmental insults, pollution, for a while vaccines until that was completely debunked. So there are these obvious things that we are concerned about, and then there's these very non-obvious routes by which cognitive development can be affected. And that's what I'm particularly interested in.
Hi, great talk. It was very interesting. I was curious, and you alluded to this a little bit, but in the different neural networks that are suggested, or the different regions that are activated in boys and girls, do you think there would be more overlap in those? Is it your expectation that there would be more overlap if you kind of took a broader kind of lumping approach to the psychiatric conditions, the outcomes that you mentioned, like depression, anxiety? Or has it been shown that those differences are maintained?
We don't know yet. I think there will be more overlap, but not necessarily for the reason, like the obvious reason. I think that there will be a common set of brain systems that are affected, and the story is going to be a lot like genetics. So in genetics, each area of neuropsychiatry, kind of has a path that they're following, and early heritability studies, GWAS studies, looking at rare mutations of large effect, and then coming back to follow up with the common variation. And what seems to be converging is that there are a set of genes that reflect risk for any neurodevelopmental disorder and many neurodevelopmental disorders. And it's really about the story of the timing of development that helps to predict which neurodevelopmental disorder. But a lot, we have, I think, 100 and something candidate genes for autism now, but none of them are specific to autism. And you can think of the imaging data in a similar way. There are candidate systems that are predictive of autism, but no one is specific to autism. I think over development, the brain systems that are affected from living with autism will get more and more specific, but that's a normative developmental process. The overlapping core systems that reflect risk for neurodevelopment, I think, are mostly going to be overlapping, and that will be true for boys and girls. And then the boy-girl autism difference is going to be another reflection of developmental processes, and it might as well be the difference between schizophrenia and autism, as far as kind of the same approach to the problem, but it's just that they happen to be, behaviorally we see them as the same thing. And then, what is that underlying set of brain systems, and how does it differentiate that predicts the different types of conditions? That's where I think the overlap will be.
So my background is in cancer biology research and neuroscience. So I want to ask a question that kind of relates how I see cancer research, which has been going on longer than neuroscience, neurodevelopmental disorder research. Can you hear me? Is this working?
Yeah.
Okay. So within cancer research, there's something called the hallmarks of cancer. There's this large word, cancer,
Yep.
but for those in the field or those with experience personally, that's a general word.
Mm-hmm.
And it seems to me that we could say neurodevelopmental disorders would be the equivalent level meta word here.
Absolutely.
Okay? So my question is, thinking about the genetics. So in the study, in the genetic research, in cancer research, so there's the history of it and how the genetics of very specific types of cancer were researched specifically, and then the different individual specific to very specific cancer lines of research then started to kind of do this with each other,
Yep.
and then develop a theory, you know, the two-hit theory that the thing about you said about the genetics of girls
Yep.
kind of made me think of. And that's the introduction to the question.
Yep.
Okay? So the question is, where are you at, where is the field at now relative to creating, and I want to ask this in a way so I'll get the answer that I'm actually looking for. So give me a second, okay? So I think what I'm asking about is, where are we at in terms of a unifying conceptual basis for research that will allow the specific areas of research within neurodevelopmental disorders to both be for the data collection to be reasonably consistent enough for there to be some basis for comparison.
Yep.
But more than that, to be able to go, you know, in and out of neurodevelopmental disorders, which go across all the categories that we name in the DSM, for example, as you know.
Right.
Okay, so that,
Yep.
you understand what I'm
think
asking,
so.
right? Okay, thank you.
So, in our field, I'm going to turn the question around, kind of ask a rhetorical question. If I said to you, you know what I really think is limiting progress is that we don't think of cancer as a spectrum. And we really need to think of the spectrum of cancer and autism spectrum, or cancer spectrum disorder, because then we'll really be able to start developing heterogeneous groups of people. I think most cancer biologists would look at me like I've lost my mind. No? The cancer spectrum. So it's all one thing, but it's a spectrum of severity.
That's where I'm coming from related to this piece. So
Right. So I'm saying that from my naive understanding of cancer research, that the idea that there were different cancers and different mechanistic ways to get to cancer which described an abnormal pattern of growth, from kind of my simple.
That's good.
And
an abnormal pattern of growth, you can say in neurodevelopmental
right,
disorders is abnormal.
it's going to be an abnormal brain development. Yeah, and we're fortunate that, roughly speaking, we can talk about the same organ, the brain. But even there, you know, multiple different brain systems. I think that on the one hand, you've seen kind of an RDoC attempt, which says, let's look at different vectors of behavior and take different patterns of behavior as the way to slice up the pie of neuropsychiatry and study mechanisms there. I think that's not a very good run at autisms. And then I think we've got, in autism, we're still trying to define that broad level and what's autism. And we're coming at it from the point of view of, well, it's this pattern of behaviors. And in point of fact, for researchers, it's a score on 2 measures and a clinical opinion. And in the presence of a cutoff, then you have the thing of interest to study. If I did that with cancer, I think I would be artificially, restricting the field to a point where it would be like saying the only measurements that we're going to count are whether you can push in on the organ that's affected and feel a lump. And that's your definition. Versus applying all the molecular tools, microscopes, everything you've got to characterizing that abnormal pattern of growth. And very quickly, instead of the presence of a lump somewhere, let's characterize that pattern of growth as the phenomenon of interest. And at least now we're quantitative. And so, in realizing I'm being kind of unfair to the instruments, it's better than a lump, presence or absence. But to get into the cancer study, if you had to have a lump or not a lump, you were excluded, then that would be a problem. And I think that's where we are in autism.
I'm interested in emotional regulation and that slide where you showed the difference between boys and girls. And so I'd like your thinking on this because for years, many developmental psychologists and some neuropsychologists have thought that emotional regulation should be a factor in diagnosis and in the DSM. So what's your thinking on that? In autism specifically, yes. There's even an instrument that Sam Goldstein developed that looks at that.
I would agree, especially as a parent. It's the number one issue that all parents bring up. And then when you're talking with young adults with autism, it's the first issue that's brought up. That the kind of experience of regulating one's emotions and having difficulties with that is a prime factor across boys and girls, and really should be core to the diagnosis. And I think I like that idea. Having said many critical things about the behavioral level of diagnosis, so you have to kind of take my view of that with, I don't know that it would fix much, because I'm arguing for a more brain pattern of development based diagnostic system. But I think it would be helpful in terms of really kind of understanding the phenomenon. And also figuring out, are the emotion regulation difficulties that we're seeing in autism, suspending boys and girls for a minute, are they the result of these differences in social being and learning to regulate emotions, and or are they kind of there immediately and part and parcel of the problems in social functioning? And you can imagine that in some people one drives the boat and the other follows, and then vice versa. So I don't know, but I think that it should be a very active area of research, and I would be all for having it as a core aspect or one of the dimensions along which you're describing people with autism when you're making the diagnosis.
Well, thank you. Thanks everybody for coming, and we hope to see all of you at our annual Autism and Public Health lecture next month.